Right, okay, it's 1 p.m. I think we will get started with today's lecture. So in the first couple of lectures, we learnt about DNA structure, we learnt about DNA replication, and today we're going to learn about how DNA can be mutated. We're going to learn about what are the consequences of these mutations, what causes these mutations, and we're going to learn about how DNA can repair itself. So, like I mentioned to you, the two textbooks that I really like and I think explain things really nicely, including the pictures, and I've got many other pictures here, is Molecular Biology of the Cell, but also Essential Cell Biology as well. So again, like I mentioned on Friday, I really recommend that you consolidate your learning by reading what's in the textbook as well as these lecture slides. So after today's lecture, you should be able to explain what a mutation of DNA is. I'm going to give you a definition. Definitions are really good exam questions. So make sure you know what a mutation of DNA is. Describe the processes that result in the mutation of DNA, and we're going to talk about these, such as chemical, UV radiation, infection, and we'll touch on all of these. Describe the consequences of depurination, deamination, thymine dimer formation, and double-stranded breaks on DNA replication. Understand how transposable DNA elements, transposable DNA is a sequence of DNA that can be quite large, they can be relatively small, but understand what they are and understand how do they mutate DNA. Infectious agents, we touched on this in my first lecture already. We know that some infectious agents, some viruses, their DNA or can get integrated into our own DNA, and that is a permanent change to our DNA. So understand how some infectious diseases can introduce mutations into DNA. And then we're going to talk about the two mechanisms of DNA repair. The first is mismatch repair system, and the second is homologous recombination. So these are the learning outcomes of this lecture. So, definition of mutation. So mutation is any permanent and heritable change in the DNA sequence of an organism. So the words that are important, permanent and heritable change. So once the DNA has been changed, you're passing that on to the next generation or the daughter cells. Consequences of mutations of DNA. Damaged DNA will cause problems with DNA replication because remember I was saying to you that the machinery that's required, the protein machinery that is required for DNA replication recognizes the DNA strand, and so if there's any distortions on the DNA strand, that can offset proteins binding from it and being able to function. But we will go through that. Repair restores DNA replication. It restores correct nucleotide sequence. Repair, although repair can also result in incorrect nitrogenous bases being incorporated. However, if you remember, I said DNA polymerase has proofreading ability, which means it can check for these errors. So to overcome these problems, all living cells have mechanisms for DNA repair. It would be a disaster if they didn't. So just a reminder, I know you've seen this quite a bit, but just as a reminder before we go on to the rest of the lecture, here is double-stranded DNA. We have anti-parallel strands. One go 5' to 3', the other one goes in the opposite direction, 3' to 5', anti-parallel. And then you have this complementary base pairing in the middle, which is really, really important, especially for the fidelity of the DNA. G hydrogen bonds with C and T with A. And then this double-stranded DNA shown here as the double helix. And just a reminder, here is your minor grooves, and here are the major grooves. So you can imagine if one of these bases were mutated, then it would create a distortion that would interrupt DNA replication. So how do changes in DNA sequence, how do they actually happen? Well, the replication errors are actually very rare. So this is why DNA replication is referred to as high fidelity. DNA polymerase, the incorrect copying by DNA polymerase results in only one error in 1 billion bases. It's very accurate, just one error in 1 billion bases, or 1 times 10 to the 9. And the reason for this high fidelity is because of what I've just mentioned, which is the base pairing structure of DNA. The fact that DNA polymerase requires a primer. Remember the primer where DNA polymerase attaches at the 3' end nucleotide residues, and the primer is synthesized by, can anyone remember? Correct, DNA primase, which is actually an RNA polymerase, but we call it also DNA primase, synthesizes the primer, and DNA polymerase comes along and attaches base pairs at the 3' end. And then there is this proofreading of DNA polymerases. So we'll have a look to see how that works. So here we've got the double stranded DNA. In the yellow, all the pictures are the same. They're either from molecular biology of the cell or essential cell biology. In the yellow is the parent strand. You can see here the bases are a string of A's. And then the daughter strand is in red. And so you can see that the first 4 bases has been inserted correctly. You've got T, 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 T. But then somehow DNA polymerase has tried to attach a C, and C does not base pair with A. So it doesn't base pair with A, creating these distortion. So this unpaired 3' hydroxyl end of primer blocks any further elongation of the primer strand by DNA polymerase. So DNA polymerase can't bind to this stretch of DNA because of the distortion, and therefore can't attach the next nucleotide. So what DNA polymerase has is this 3' to 5' exonuclease activity. And what this activity does is it basically chews back to create a base paired 3' hydroxyl end on the primer strand, like so. So it chews back and gets rid of the cytosine. And then DNA polymerase continues the process of adding nucleotides to the base pair because, of course, it's chewed back the incorrect base pair, so now it can bind to and attach to the DNA, and it can start adding the correct base pair again. So this is how the proofreading capability of DNA polymerase works. So mutations of DNA, there are many environmental factors that can mutate DNA. And here I've got this picture. Again, it's of DNA, but it's just a single strand of DNA. So at the bottom is just shown slightly differently. You've got the sugar phosphate backbone. And then you have the bases here, G, C, T, and A. Now, what I've shown here are the types of attacks that DNA can get. We've got methylation, we've got oxidative damage in the pink, methylation is in green arrows, oxidative damage is in the pink arrows, and then we've got hydrolytic attack with the blue arrows. And the thickness of the arrows shows you how frequent these attacks are. Now, if you have a look at this figure, you can see that these arrows are absolutely everywhere. So you can see, even in the sugar phosphate backbone, as well as the bases. So you can see how susceptible DNA is to these environmental factors and attack. So we have the three categories of environmental factors. We've got chemical, which can introduce nucleotide instability. There we have mutagenic chemicals, and I'll give you some examples shortly. Radiation, which can, especially UV radiation. Here in Australia, we know that the UV can be quite dangerous, so we're told to put on a sunscreen. Well, UV radiation can result in mutation of DNA. And also other types of radiation, such as gamma rays. And then we have what we call mobile DNA. And here, this mobile DNA, can be caused by infectious agents. For example, the HIV example that I gave you. And transposable elements. Transposable elements are stretches of DNA that can transpose themselves basically onto different chromosomes. And again, we will have examples of these. So the first example of mutation of DNA is nucleotide instability. Now, here we've got depurination. What are the purines in DNA? Clues here: guanine and it's the two rings. So guanine and adenine, they're the purines. And so what we call depurination is when basically a purine is lost from the DNA backbone. So here we've got the DNA strand, the sugar phosphate. Here is guanine. Guanine is lost, and you just have the sugar phosphate after depurination. You can also get deamination. I would make sure you learn what depurination and deamination means. And deamination is a loss of an amine group. So in adenine, when the amine group is lost, you get hyperxanthine. Guanine, when the amine group is lost, you get xanthine. Cytosine, when the amine group is lost, you get uracil. So but thymine doesn't have an amine group, so there is no deamination. Now you can immediately see that if the cytosine undergoes deamination to a uracil, you can see how it's changing the genetic code already. Any of these, hyperxanthine or xanthine, it changes your genetic code. So let's have a look at deamination and depurination and the consequences of them. So here we've got the parent DNA in yellow, and the C, which base pairs to the G, has been deaminated, and so it's converted to a U. So from the previous slide here, cytosine, when it's deaminated, is converted to a uracil. So here, during DNA replication, if you remember, both strands are used as templates for synthesis of the new daughter strand. So let's see what happens when you use the bottom strand as a template. So we've got A, G, T, A, and then the new daughter strand, which is in the red here, and the bases are T, which base pairs with A, C to G, A to T, T to A. And you can see that this strand remains unchanged. It's exactly the same as the parent strand. However, if we take the top strand and use that as a template, then you have T, U, A, T. DNA polymerase will come along and attach an A, which base pairs with T, an A that base pairs with U in RNA as well, and then an AT and another AT here. So you can see how this has changed its code. The one, that deamination event, has changed the code of the newly formed DNA. Now with depurination, this is where you see a loss of a purine. Here, the A has been lost. So here, if we do exactly the same as what we did for the first example, if you take the parent strain and use that as a template, so we've got the AGTA, then the daughter strand is TCAT. It remains unchanged and it's exactly the same as the parent strand. However, if we take the top strand now, so here it is, that's the TC. The A has been lost, so you've got a T here. And then the new strand, AGA. So you can see that this is different to the parent strand. So the outcomes of this is that DNA polymerase randomly assigns nucleotides to match the damaged nucleotide. So here it's just, it's not added anything here because it's lost it and moved on to the next one. And here it's added an A because A base pairs with U. So DNA polymerase randomly assigns nucleotides to match damaged nucleotides, and this changes the nucleotide sequence. And when it changes the nucleotide sequence, this becomes fixed and inherited. So mutagenic chemicals. So I'm going to talk first about alkylation. So alkylation, electrophiles add alkyl groups to nitrogenous bases. So an example here is methyl methane sulfonate, or MEMS or MMS. And you can see here is guanine here, and the guanine's been alkylated and it's converted to N7 methylated guanine. And so this will create a distortion, and DNA replication will not be able to occur because our DNA polymerase won't be able to recognize it. And other examples of chemicals, there are other carcinogens and methyl methane sulfonate. And then there is intercalation. And the example I'm going to give you here is of ethidium bromide. Ethidium bromide used to be used quite routinely in the lab to visualize DNA. I used it during my PhD. And so we used to use it to visualize DNA. Ethidium bromide is a carcinogen. We don't use it anymore in the lab. We have much safer options now. And so this is what happens. Ethidium bromide actually integrates into the DNA and intercalates it. So here is a double-stranded helix here. You can see this is looking nice with the minor and the major grooves. And then this is DNA with ethidium bromide intercalated in. And you can see how it distorts the double-stranded DNA. And this will also stop DNA replication. So that's mutation of DNA through chemicals. And then UV light. So this is the picture that I've got at the front of my lecture today. And UV light can cause the formation of thymine dimers. So here is the DNA. You can see there are two T's next to each other, thymine here, and UV radiation causes dimerization of these thymines. So the thymine bases become covalently attached to each other, forming thymine dimers. Now you can imagine in a long stretch of DNA code, there's going to be plenty of T's that are next to each other. So this can cause a lot of problems. So damage in DNA is caused by exposure to UV light. So here is UV light incoming, and here is the formation of thymine dimers. You can see the distortion in the helix that it's causing. So other forms of radiation also causes damage to the DNA. These include gamma and X-rays, and they attack DNA bonds by directly producing free electrons, which attack DNA backbones. So here you can see that happening over here, free electrons that attack the DNA backbone, or indirectly, as shown here, by generating hydroxide free radicals, and this also damages the DNA. These hydroxide radicals here also can cause damage. And it can cause damage at a single strand, or as shown here, it can cause damage at a single strand, or it can cause damage in both strands. So then the repair mechanism, you would either have to repair the single strand, or if it's a double strand, then that would have to be repaired as well. And we'll talk about how all this, how the repair mechanisms happen at the very end. So then there is mutations of DNA caused by infectious agents and transposons. So this is also known as mobile DNA, and it has the ability to insert or recombine into target DNA molecule. Examples are infectious agents, for example, viruses and bacteriophages. The example that I gave you was HIV. HIV is an RNA virus, and the virus also encodes for the reverse transcriptase enzyme, which converts RNA to DNA, and that DNA is then integrated into our genomes, causing a inheritable change, a permanent inheritable change to the DNA. It's not just HIV, but COVID as well. That's an RNA vaccine too. So it's not all viruses, it's some of them. And also bacteriophages. Everybody know what bacteriophages is? Yep, excellent. So they're viruses that attack bacteria. And then we have transposons, and these are mobile genetic elements that can jump from one chromosome to another, or from one DNA strand to another, and they were called jumping genes. I've got a picture here of some maize here, and this is where a scientist called Barbara McClintock was a geneticist in between. She did a lot of work on these jumping genes. She discovered them, and she did work on maize. So maize naturally has a lot of different colours, and this is because of this transposons jumping from one chromosome to another, inducing change in colour. Barbara McClintock, when she came up with this hypothesis in the 1940s and 50s, and she tried to publish the data, there was a lot of scepticism in her studies, and people didn't, and she was finding it very difficult to actually publish her work. It wasn't until the late 50s when other scientists were discovering the same thing of these genetic mobile elements, these transposons that are jumping, did her hypothesis get accepted. And she got the Nobel Prize for it in the 1980s. So this is these transposons and these mobile DNA from viruses and bacteriophages undergo the process of recombination. So recombination is the breaking and rejoining of DNA molecules to form new combinations. So I've got this picture here. This is an example of what can happen. So here we've got chromosomes, and these chromosomes are homologous. Do we know what homologous means? Homologous means it has a high sequence identity, or they're exactly the same, or they're very, very similar. So they're pretty much exactly the same. And so what you get is you can get a crossover of these chromosomes, this part over here, and then what happens is you get combined genetic information. So you can see that this chromosome has a bit of red and this one has a bit of blue. So non-homologous recombination means there is no similarity between DNA molecules is required. So this can happen between chromosomes that have no sequence identity, so they are non-homologous recombination. But other things are needed in this instance. You need the site-specific or targeted recombination, which is catalyzed by enzymes called integrases and transposases. And I will talk a bit more about that in a minute. And then homologous recombination, we've already gone through this, means both the donor and acceptor DNA molecules have extensive similarity in DNA sequences. So that means if they've got extensive sequence similarity, they can act like a template for each other during DNA synthesis. So, infectious agents. Well, retroviruses such as HIV, which causes AIDS, and their equivalent in bacteria, such as bacteriophages, can integrate into host DNA. So here is a viral RNA. You can see its RNA represented in the green. And then this blue box here is reverse transcriptase, the enzyme that converts RNA to double-stranded DNA. So the virus infects the cell, it gets in, it releases its RNA, releases its reverse transcriptase, and undergoes a process of reverse transcription. And they're essentially parasites. They use our host cell replication machinery to do this. They can't do it on their own. And so through the process of reverse transcription, you get viral DNA, and then this viral DNA integrates into the host DNA. Now, there are two types of bacteriophages. There are ones that we call lytic, or two types of viruses. Ones that are lytic, basically they enter into the host, and when they've replicated, they lyse the host, killing them. And then there's what we call lysogenic. Now, lysogenic integrate into the host chromosome, but don't kill the host. And so this insertion of this foreign DNA can disrupt coding regions. So coding regions are the genes, and the genes are what codes for, eventually, the protein. Now, if you've got DNA that integrates into the gene, then you are going to disrupt that process of protein translation, which means that you will get an incorrect protein which could harm you. Now, some viruses are very clever. HIV, for example, it prefers to integrate into transcriptionally active genes. So basically, it means that it knows that its DNA will get transcribed and then will get produced into protein. So that's one way of it ensuring that it's always going to be able to replicate. Now, it's not a common feature of all viruses. Only some viruses do this. So transposons, or jumping genes, these are basically linear DNA molecules that move within and between chromosomes. And they insert into many different DNA sequences. So I've got this quite simplistic figure, but I think it illustrates the point really quite nicely. So here we've got DNA A, which is in the darker grey, and it has a transposon here, inserted in here in the green. And then you've got DNA B, which will be the recipient DNA, and so the transposon literally jumps from one into another, as shown here. And in sometimes, and it's not shown in this figure, but sometimes it jumps and it leaves DNA A, for example, and goes into DNA B. Other times, it can replicate it. So it can use it as a template and replicate it. So then both DNA A and DNA B will have that transposon then. So what's the consequences of this? Well, insertion, again, same with like the viral and the bacteriophage DNA, insertion of the transposon could physically disrupt genes if it went into a gene. Excision of the transposon can result in small duplication of DNA. Again, this will disrupt the gene. And but it is common in bacterial DNA rearrangement. So I'm going to give you the example of, sorry, I'm just going to have a drink of water. Sorry about that. So, bacteria in the soil. AMR, antibiotic resistance. Bacteria in the soil are surrounded by lots of different bacterial species. Some of them have antibiotic resistance cassettes. And using this method of transposons, if the antibiotic cassette is encoded in transposon, that transposon can jump from one bacteria to another, meaning that it can transfer its antibiotic resistance to another bacterial species. That's how bacteria is becoming more and more antibiotic resistant. So, now I'm going to talk about methods by which a transposon can go from one chromosome to another. So here we've got chromosome A in the green, and here we have the transposon in the yellow. And this transposon is going to jump from chromosome A to chromosome B. And so you can see that the transposon has these this red, what we call short inverted repeat sequences on either side. And the transposon encodes for proteins such as these transposase monomers. And so these transposon monomers is what helps this transposon jump from one chromosome to another. So we've got, so what happens is the transposase monomers attach just outside of these short inverted repeat sequences. Remember, these sequences are signals of where the transposase should bind to. So once it's bound to it, it forms what we call a transpososome. It loops in on itself, and it is excised. And this is a central intermediate. Along comes the target chromosome B, which has these short direct repeats of target DNA sequences. And these short direct repeats tell basically this intermediate where to insert itself. And so it gets integrated into chromosome B here. Now, in this example here, so we've got what happens to chromosome A. Well, there is a double-stranded break in chromosome A. So we have repair mechanisms that come into play, and these repair mechanisms rejoin this donor chromosome A, fixing it. So this transposon has jumped from chromosome A into chromosome B. And in this instance, it's a cut paste, and so there is no sequence homology required because it is a cut and a paste. So this is the example that I've just given you here. So this is a non-homologous recombination. So this is when the two chromosomes, the target and the donor chromosomes, are not similar. So you have the cut and paste, non-replicative transposition. So here is the transposon in red, the donors in grey. Here's your target DNA in yellow. So you get the cut and the paste done by these transposase enzymes, which is actually encoded on the transposon itself. So it carries the machinery it needs to in order to do this process. And so it's cut out of the donor and enters into the target DNA sequence, as shown here. Now, you can get also replicative transposition. So this is where the target DNA uses the donor DNA as a template. And so it uses it as a template, and it acquires the new DNA sequence, and both the donor and the target have the transposon. Now, you might be wondering how it can do this. It's quite a complicated process, and you don't really need to know it. But essentially, the two DNA actually link together, and then one acts as the template for the other. So, non-homologous recombination. I'm going to give you the example of bacteriophage, and I'm mentioning this bacteriophage T4 because this will be in your lab too. So don't forget that you'll have some pre-lab reading material and a quiz to do, and please go and have a read of it. But basically, in lab 2, you are going to be given some bacteriophage T4 DNA, which is circular, as shown here, and you will be doing some restriction digest on this DNA and running it on a gel and doing some calculations on it. So I've got this lovely EMs. You know, I do like showing you EMs. I think they're pretty cool. So here you've got E. coli, and you see this T4 bacteriophages in the white attaching itself, attacking the bacteria all the way around it. So, the bacteriophage T4 has sites known as attachment sites. So these, and it has an integrase, and this integrase is a protein that promotes its recombination into specific attachment sites. Now, in E. coli, so this is the bacteriophage DNA, circular DNA, and this is E. coli chromosome, and you can see that basically E. coli has these attachment sites. The E. coli genome has attachment sites as well that is recognized by integrase, and so then integrase inserts the T4 bacteriophage DNA into E. coli, like so here, into these attachment sites. So that's how integration of bacteriophage T4 into E. coli can work. So I've talked about mutations, we've talked about consequences. Now we're going to move into DNA repair. How does DNA repair itself? So there are two examples that I'm going to give you. The first is mismatch repair system, which repairs mutations in newly synthesized DNA strand to restore it to the original sequence of the template strand. So mismatch repair systems happen in combination with DNA synthesis, so it's on newly synthesized DNA strands. And then we're going to talk about homologous recombination. Homologous recombination is required to repair double-stranded breaks in the phosphodiester backbone of the DNA. And you can get two results of this. First of all, you can get restoration of the original sequences, but you can also get rearrangements of local regions of the DNA sequences as well. And I'll show you this as an example. So DNA repair, this is basic mismatch repair. But before we go into mismatch repair, this is how important mismatch repair is. So if we had DNA replication without mismatch repair, you would make one mistake per 10 to the 7 nucleotides that are copied. If you have mismatch repair with it, DNA replication, you only get one mistake per 10 to the 9. So you can see without mismatch repair, there's 100 times more likely to get mutations in your DNA. So that's how important it is. A mismatch refers to mispaired nucleotides. And repair, you have these special repair proteins that recognize and excise the strand of DNA containing the mismatch, and then DNA polymerase, repair DNA polymerase. Remember, we talked a lot about the polymerases in my last lecture, and in E. coli we had two DNA polymerase 1 and 2 were repair DNA polymerases. DNA polymerase 3 is the one that does most DNA synthesis. So there's essentially 4 steps. So the first, so here is your double-stranded parent DNA. The first is here is that the DNA is damaged. So you can see an incorrect base has been attached to the DNA. So first you have these repair proteins that patrol the DNA, looking for these mismatches, looking for distortions in the DNA, and then they bind to the mismatch sequence. The mismatch sequence is then excised by nucleases. Nucleases degrade or chew the DNA, and it creates a single-strand DNA patch over here. So then, synthesis of the second strand by repaired DNA polymerase, so the repaired DNA polymerases, using the free 3-hydroxyl group as a primer. So it uses this as a primer to repair this damage here. And then once it's added the correct base, DNA ligase comes along and seals that nick. So that's how DNA mismatch repair works. So repair of DNA by homologous recombination. Remember I was saying that homologous recombination can result in exactly the same parent strain, or it can result in changes. And I've got some examples here. This is mainly in meiosis, and you've got 2 chromosomes, red and blue, and you can see that the red and blue have crossed over and exchanged DNA, as shown here. Now, these chromosomes, the new chromosomes, then undergoes meiosis, and you can see that there are 4 possible gametes. You can get a full parental red, which is exactly the same as the original here, or you can get a full blue, which is the same as the original here, but then you get these recombinant genotypes. Now, this happens in nature all the time. It's natural, it gives the diversity that we see. And so these regions have very similar sequences. So remember, homologous recombination, the DNA is pretty much identical with just some changes if there is any. So the regions are very similar sequences, and these sequences align and can result in crossover of the DNA. So the double strands are broken, then a crossover occurs. DNA repairs, generates new combinations of DNA as shown in this example here. So this is how homologous recombination works. I'm going to go through this slowly. It can take a couple of read-throughs to understand it. Again, I recommend that you read Albert's. So I got this from the 6th edition. You've got access to this in the library. This is from Essential Cell Biology. So first of all, we've got 2 DNA molecules. We've got the yellow here and the red, and they are homologous. So their sequences are pretty much identical. So there is a double-stranded break that's been introduced to the yellow chromosome here. And so first of all, what happens is that you have these specialized nucleases, also called exonucleases, and they digest the 5' ends of the broken strands. So you can see on this strand here, this would be the 5' end on this strand. It's been chewed back. And then at the top, this will be the 5' on the top strand, and that gets chewed back that way. Okay, so choose these DNA at the 5' ends. Then what happens is what we call strand inversion by complementary base pairing. So these are now single-stranded 3' overhangs. Yeah. There's a 3' overhang there, and there's a 3' overhang here. And so the single-stranded 3' overhang migrates into the recipient chromosome in the red, where the sequences are the same, so where the sequences are homologous. So here you can see that this bottom strand here, this 3' end, has migrated or overhanged, invaded into this red chromosome, and complementary base pairs with this because the sequences are the same. So then what happens is repair polymerases, so DNA repair proteins, synthesizes the DNA where it's been damaged here in the green, and so it's synthesizing it using the red, which is undamaged complementary DNA as a template. Okay, so now I've got a second part to this, but this figure here is exactly the same as this one. Okay, so so here we're getting the synthesis of the strand using the red chromosome as a template. So then these cross strands, when these cross strands happen, we call them holiday junctions, named again after the scientist that discovered it. So rotation of these cross strands allows section of one strand to be joined to section of another strand. So you get exchange of DNA. So basically, once the newly synthesized DNA, the bit that's been damaged, has been newly synthesized and repaired using the homologous strand, the red chromosome, you get exchange. So it comes back here to join up with the other part of its yellow chromosome, and then DNA synthesis continues using the complementary strands from damaged DNA as a template. So you get, so this has been, this continues because it's homologous now. It's using this as a template that hadn't been damaged, and the same with the top strand here using this as a template. And then you get DNA ligation. So DNA ligase comes along and nicks the gaps together. And then you get nucleotide sequence at the site of exchange is unaltered. So there is no additions or subtractions. So that is how a double-stranded break is accurately repaired. So if you want me to go through that again, I'm happy to go through that. Just let me know. I can do that in the next lecture if you like. Or if you want to come and go through it with you, then just come and see me after this lecture. So I've included this background reading. So molecular biology, the cell fifth edition, essential cell biology, that is where the homologous recombination figure comes from. So I recommend you read that. And I've got you some other stuff here for your own interest. Thank you.